a question during the break. That way we do have to do the smoothing. And I'm sure I gave the right answer, but it seems she got it. But I will just give you my explanation of that. Okay. As I mentioned to you before, that my background was electrical engineer. I was a control engineer, and we measure the electricity in the motors, the rotations of those. And these type of signals are something that we see a lot, which means that you see there's some of the real trend of signal. That is what real signal look like, together with many noise that's sitting on top of it. Okay. Sometimes it depends on the distribution of those noise. This is a side issue. We're just waiting for people to come back. Right? So when you see these signals, when we look at the different noise distributions, and there's something called the white noise. That's something we see a lot, which means they really don't follow certain patterns, but their intensities are kind of just random and just. So what can cause that is once you do the measurement, your equipment may receive some kind of random noise from somewhere, and then that on top of it, if your signal is small enough and noise is bigger enough, and they cause this type of thing. For this scenario, and those noise can be caused by, for example, our sampling issue. So the sequence is not deep enough, so for that region, and you got more rays or versus not too many rays, and those are just a random sampling issue. If you sample deeper enough, and you probably get the noise smoothing out a little bit. And also it's possible to caused by the mappability issue or other type of a chemical issue. For example, when you do the sonication where the breakdown should happen, or especially when you do use enzyme digestion where those breakdown could happen. So there's a lot of random effect happens. And that's why that you see this type of signals. And if you see there's a big peak around here, and then there's a follow with a sharp dip, that really doesn't mean anything. It's just a noise on top of the real signal. So once you do this window size smoothing, and that will potentially help you to recover the original signal in a much greater detail. Okay, that was my explanation. I'm not sure she bought it, but it seems that she got it. All right, but anyway, so let's move on to the next topic, which is the background issue. Okay, and so once in the previous one we talked about the tag shifting, tag extension, and the goal of those is all trying to identify the signals from the ChIP-seq data. But if you see there's a peak around here, if you want to say, is this a good one? Is this an enriched one, ChIP enriched one? If you only look at this particular peak, you cannot make any conclusion. Okay, if we do not comparing this with the background signals. And so what is the background signals? The statistical significance of tag clustering depends on the expected background pattern. One assumption that most people make is background tag density is distributed uniformly along the genome and independently between the strands. So you can see this is one example. So you see the blue one is a ChIP enriched signal, and the black one is the background signal. Okay, and for this one you can clearly see this background is not keeping up with the ChIP enriched signal. So you can have certain level of confidence to say this is a real ChIP-derived peak, enriched peak. Okay, but if your background at this location happens to be also the same type of pattern, and you probably won't define this as a peak. That's why I say that once you want to calculate the statistical significance, not only you want to see what the signal look like, but also the background look like. And the source of, there's many different sources of non-binding enrichment. Non-binding enrichment. So this is not a non-binding enrichment. This is the binding events. But if in the input sample, which is non-binding, and you also see this type of enrichment, that also happens a lot in the real data. If you look at the ChIP-seq data, and for some no IP, no immunoprecipitation control samples, sometimes you also see this type of peaks somewhere in the genome. We see that a lot. there's many different ways that this can happen. The first one is there's a variations in the chromatin accessibility. So we know that some part of a chromosome, they are packed very dense. So it's really hard to break it and to further do the sequencing. But some other part is very loosely packed, or there's no histone around that region if it's an extended open area. So even though you don't do the protein enrichment, the ChIP enrichment, you will still, it's easier for you to grab that part of signal. So sometimes you see, even in the no IP input control, you still see a lot of enrichment here and there, and this is one of the reasons for that. Another one is more like a sequencing-related variation, that this will include the mappability issue. Maybe some region nearby region you don't see any signal because it's not really mappable, and or some of the sequences are harder to sequence than others. For example, if one sequencing raises all the CG, 90% are CG, and it's pretty hard to sequence that fragment. So if you've got more AT, you can. So those kind of sequencing composition differences can also give you this sequence-based variation. Okay. So again, the point of this slide is once you want to study the significance of a peak, you better look at what the background is. There's a couple of different types of background noise. So this is a paper, I think is cited in the previous slides, and by Peter Park's group at Harvard. So you can see, if you don't see it very well. So there's actually two lines in this figure. One is this dark, this really black line. That is the input sample. And another one is this, a very faint one. That is the ChIP enriched sample. So the first type of variation, you will see this singular peak of tag density. And their conclusion is that if you look at this type, it's pretty easy to get rid of. So you see there's a kind of enrichment in the ChIP, and also there's an enrichment here in the input samples, and that suggests that this region has something weird and going on. So you can really get rid of it. The second type, which look like more like this, is it's non-uniform, but it's really, really wide. As you can see, there are over 1,000 base pairs. You can see this, and the clusters of increased tag density on one or both two strands. So actually, there's also the ChIP and the input samples in there. So this is another type of background noise. You can anticipate a lot. And the third type, which is really difficult to get rid of, is once you look at the reads, it really looks very much like real. If you have a good eye, especially when you see it from the back, there's a faint line here. The faint one is the ChIP enriched ones. So you can see this is almost identical. It's a perfect example as what we see from previous slides. However, when you look at the input samples, it also has this pretty perfect shape as well. And that suggests that this region has some problem. And for this type of regions, you really, really need an input control to get rid of. If you don't do input control for this, and probably you don't get rid of the noise. So this type 3 is normal peak, but appear in both control and ChIP enriched assays. Very difficult to handle, but need negative control to get rid of. Okay? But point here is people studied, and actually this study is based on this 2007 Science paper, the first ChIP-seq experiment, and they derived this several different type of background noise. And to call a peak, we need to construct a background model. Again, when you look at this, so you see this is the real ChIP enriched signals. There's a peak here, and this is the background signals. And how do you estimate the background signal? So there's three different scenarios. The first one is, remember, a lot of people, to save money, we do not do input control. And there's one scenario which is no control. And the second one is you do have a negative control. And the third one is you want to compare two different conditions before and after drug treatment. So you do not have input control for this, but you do have two different conditions to comparing against each other. For example, MCF7 versus MCF7 treated with estrogen. And that is a two comparison. And this is one sample, MCF7 versus MCF7, the input control. And And the first scenario is there's no control at all. So let's get through the first one, which is no control. What people do to call it a peak, which they will estimate the background tag distribution using a Poisson or negative binomial distribution. So if they observe a peak around that region, okay, they will, under the assumption that all the background noise should be equally distributed, and one window size they look into, that number of fragments, even in the background, should follow a Poisson distribution. And then they can generate this background distribution and comparing with the real distribution from the data and to call the significance. The second one is with control. So when you do the input control, now you can get down to more position specific. Okay, at this location in the real sample you see a peak, and in the input sample you probably also detect some signals, and you can estimate the background noise at this particular location based on the input samples, and that is increasing your power dramatically. And the third one is with control. With control is a subtract. So you got MCF7 versus MCF7 treated with E2, two conditions. Now the strategy people may take is just to subtract the signal along the genome from ChIP-seq data. So using the E2 induced one minus the signals in the control samples, and that potentially can work. And the signals can be thresholded by its enrichment ratio relative to control. So you can see this. This is the control sample. This is an input sample. This is the real signal. And this difference is they will call it enrichment relative to background, so enrichment ratio. And this is three different strategies. Especially difficult one is the first one. When you don't do any control, you have to estimate a background noise based on that. Yes. How to estimate a background? Because you have the signals along all the genome, and those are input controls. So you can basically look at the distribution. When you break it down into smaller window size, you can count the numbers in the window size, and those are input controls, right? And then you can estimate, if you follow the Poisson distribution, you can estimate the parameters in the Poisson or negative binomial. So that's help you to get it. Well, no, that is you are talking about no control, how to estimate that. So what I was talking about is to use input control and to estimate that. But for the no control ones is you will just make the assumption that most of the genome are not binding sites. So the binding site is only a very small proportion of the genome. So once you break down the genome, the rest part into smaller pieces, even though there's some piece you do see one or two binding sites in there, and there's enrichment, it doesn't really matter. It doesn't change the overall distribution too much. You can still estimate your Poisson distribution based on the data. It's just like along the entire genome, only a very tiny proportion are the real binding sites, just based on that assumption.
I think if you don't do a control, the loss of this value is not accurate.
You will have to assume that you have enough rates on that.
So do we know, for example, what is the on-off target ratio of our how many rates
are the binding sites, some of them rates are not. Well, it doesn't really matter, because when you look at the distribution, you are looking at, you use each window as a unit. Okay, if there's one window that has 10,000 rays, let it be, but it still just be one window. And then basically by the end of the day, your background distribution estimation should be based on the histogram of the number of rays in each individual window. So if you have 100 million windows across the genome, okay, and only, let's say, 10,000 of them are real signal region, those are only representing very small proportion of overall. So you still have enough information to get you through the rest of the background noise. But we can talk about that later. Let's move on. Okay? So for the peak count, the main quality criterion is either an absolute signal threshold or a minimum enrichment relative to the background, or both. So which means that if some different algorithms, they call there's a peak in different criteria. Some of them, I do want to, this part has a lot of signals, or which is absolute signal threshold, or it has to have enrichment over certain kind of background. Sometimes algorithms require both. So different software use different specifics. And there's a review here which is very, very good. Most of my figures actually is from this review. And they also concluded that the p-value from different methods are not really comparable to each other. So if you use one algorithm to calculate p-value, say this is the peak, p-value is this, and another algorithm, they are not comparable to each other at this stage. And there's no way that we can do that. So let's go through the post-detection filter. And so after you detected this is peak, there's some other information can help you to get through some of the noise. The first one is the tag directionality. So meaning that fraction of positive and negative tags, the presence of positive and negative tags left and right of a peak. So you want most of the positive tags are in the left side of the peak, and the negative ones in the right side of the peak. So this is one of the things that people do the filtering. And they also want to eliminate tags of single sites that show counts much greater than expected by chance. So which means that in some of the regions, you might see some weird thing happen, like 100,000 arrays that's stacking on one genomic location from the bottom to the up. All right. This is not a joke. We do see that. And in our first ChIP-6 experiment, we have seen one of the samples, I think over 50% of the arrays are in one genomic location. All right. The reason for that, later people figure out, is those PCR steps really mess things up, and they really aggregate things in, and eventually generate those artifacts. But those are something that why people doing this. So if you see something weird in one genomic location, don't focus on that. That most likely is early junk. And this comes to the genetics question, right? Why we should get rid of reads for both the positive and negative strand. The reads on the positive strand should be upstream of the binding sites, and those on the negative strand should be the downstream of binding sites. So you can see that for the single binding sites, and you want to see the positive peaks like this, and negative like this, and binding sites is sometimes somewhere here. But sometimes you'll also see this type of scenario, which suggests that there's a multiple binding sites in there. And those are the things I don't have a solution for that. But once you get rid of things, you probably want to do some manual job to look at whether this sequence arrays tends to cluster together. And another thing you might also want to try is for the sequence for the region like this. Once you identified this and you are ready to get rid of it, okay. And before doing that, look at the sequence features in this region to see whether it's possible there's some kind of dimerization or trimers that in these regions, because sometimes the proteins do work in that way. Okay. All right. So this is the part of a peak detection, and we will talk about the motif finding in the later slides. Okay, and now there's a second thing that we want to go through is the fragment length evaluation. Only 2 slides here. Okay, as I mentioned here is there is a distribution for the fragment size. So just to remind you where we are. You got the ChIP-6. The first step, you do the cross-linking, and then you break the DNA into smaller pieces. And then you use antibody to target that protein. Now you have that DNA sequence, which is a couple of 100 base pair long. And then you only sequence the beginning of it. All right. So this is for this couple of hundred base pair long, that DNA fragments, and this is size specific. So we can control the size of it. And this is one of the figures. I don't know whether you can see it from the back. So I got from internet, from Sage Science. So you can see that this they run this gel, and once they did this sonication, and they got the fragments, they chipped it, and they want to sequence it. And then they really run it gel, and you can see this is one thing that is around 300 base pair, and there's another around 200 base pair. And then what their practice is, you will cut this part of gel and purify the DNA to do the further sequencing, right? So that is one of the size selection step. And this size selection in the sample preparation stage, and if it's a sonication, and that depends on the intensity of the pulse and duration that you gave for the DNA samples. But sometimes it's not only controlled by that. The reason for that is further PCR steps will favor the shorter fragments. So if you have fragments, you initially think it's about 300 base pair, that's where you're controlled. But eventually, you probably, after PCR steps, you are favoring those shorter DNA fragments. So those are really hard to evaluate it just based on the gel pictures from this stage. And therefore, the size of these DNA fragments are usually determined by computational approaches, and we'll tell you how. And as the earlier slides mentioned, when you do the peak detections, why are we concerning this? Because when you do the peak detections, both tag shifting and the tag extension needs you to know how long your DNA fragments are, right? For the tag shifting, you want to shift half of the size of the DNA fragments. For the tag extension, you want to shift the entire size. And actually, the way to do this is very, very simple. So what you got here is you just do the cross correlation of positive and negative strand tag densities. Okay, you got all the tag densities on the positive strand, on the negative strand, and then you do the correlation. So you can see X here is the fragment length. Right? If the length, the average length is about 100 base pair. So once you shift 100 base pair, you are expecting to get a perfect correlation of positive and negative signals. Okay? So what they do is they will just, X here is the read length, and the Y here is the Pearson correlation of signals by tags from the forward and the reverse strand after shifting this fragment length. Are we all get it? Or not? Yes? Pause? So, okay, let's go through that again. So you got all the signals along the positive strand and along the negative strand. We know there will be a gap here, and we are not sure how big the gap is, right? We can look at it for one individual peak, but we cannot look for the genome wide. It's pretty difficult. So what you do is you shift maybe 10 base pair of this, and to see are the two signals correlating with each other? No. Another 10 base pair? No. After you get a 200 base pair, wow, the correlation of positive and negative signals are really, really good now. And that will suggest you that the peak of this fragment is 100 base pair or 200 base pair. Okay, just shifting that a little bit and then calculate the correlation. And this is the way that we use a computational method to evaluate the length distribution. Okay? You do need to write some code on that, or you pay some money and to buy the software. So Partech actually has this functionality. You just put your sequencing reads in there, and this is the figure that is generated. So you can see clearly that there is how many. This is 100 base pair around this region. There is a clear peak there. That suggests that this fragment length is 100 base pair. Question? No? But this is a very important step we have to do, because when we do the peak calling, either tag shifting or tag extension, both are using this information. Now, the saturation analysis, and this becomes start to become, most people are not familiar with this, but this becomes really critical. The question here is, the depth of the sequencing rates. So how much is too much? When I do a ChIP-seq data on, let's say, AP1, CFOS, okay, AP1 binding protein, how many reads do I really need? Do I need 1 million reads, 10 million reads, 100 million reads? I don't know, right? So will increase the sequencing depth give you more binding sites? If it's still giving you more binding sites, you probably want to get more sequenced so that you get more binding sites, right? So one way to look at this is how many sequencing reads are detectable around the true binding sites. So when you identify true binding sites, you want to see how many sequencing reads are there, okay? So you can see this is with a big dynamic range, meaning that most of the true binding sites around that, the signal you detected are pretty low. But some of them can go through a couple of hundred base reads. So some of the binding sites has many 800 reads in that region. Others has maybe 10, 5 reads. So it's a huge, huge dynamic range. It's continuous, no clear subpopulation of binding positions here. So this suggests that increasing sequence depth allows a greater number of weak binding positions to be discovered. Okay? This part, for the sites that doesn't have too many binding tags detected, and most likely they are, we call it weak binding sites. So if you increase the sequencing depth, you have better chance to find out the weaker binding sites. Okay? And the more pronounced binding sites are easier to identify. So if there's something that is really obvious, regardless of what approach you use, you can find it. And also, maybe you don't have to sequence very deep. You will be able to find it. And smaller sequencing, for those very obvious protein binding sites, and smaller sequencing depth is adequate. But when we ask the question, so the initial question was framed a little bit not very accurate. So when we're asking, how do I sequence enough? This is a saturation issue. We should always ask another factor that's kicking in here, which is how greedy you are, right? So which means how weak a binding site do I allow? I always want to see the weak binding sites, right? The more weaker the binding sites I see, if it's real, that gives me good feeling that I'm detecting all of the information. But you have to stop somewhere. So you have to define your greediness by saying, this is, I'm going to look for this strength of binding sites, and lower than that, I'm not going to care about that. So there's a definition of this is called the minimal saturated enrichment ratio, and which is the minimum tag enrichment ratio relative to the background that is saturated in a given sequencing depth. Okay? So let me give you a real example. Hopefully this will make better sense. Otherwise, again, this will not be in the quiz, but it will be in the final exam. So you still have opportunities to work through the slides after you go back and to understand this better. So you can see this is X here is the number of tags. This is 500,000 rays, 1.5 million rays. Y here is the fraction of calls that are, so you can see for figure like this, that means it's saturated. So you further increase the sequencing depth, it doesn't help you to identify more sequencing, more binding sites. Okay, but this one is larger than 30-fold of enrichment, okay, comparing to the background. So this is really strong binding sites because the binding size itself it has to be 30-fold or more comparing to the background signals. If you are looking for that strong binding sites, you don't need too many reads. You will be able to reach the saturation. And this dashed line is a 7.5-fold enrichment. So if you are doing 7.5-fold enrichment comparing to the background, if that's your cutoff, and then you probably need a little bit further to get to the enrichment. And this will, the saturation for this definition will be 99% of peak agreement when you reduce the data set by 100,000 base pair, 100,000 rays. Okay? So you probably need to think this through a little bit. But point is, when you evaluate the saturation, you want to have a better idea how strong your signals, binding sites you're targeting. If you're targeting very weak binding sites, you want to sequence a lot deeper. If it's not too weak, a 30-fold difference, and you got only 300,000 rays, you probably already get to the saturation. That make sense? All right? Just a concept-wise. And this is just a different way to look at this. This is X is the log of number of tags, the sequencing depth, and Y is the log of your greediness factor, right? And you can see clearly that if you are becoming more and more greedy, meaning that you're targeting the weaker and weaker binding sites, you start to need more and more sequencing rays. Actually, I want you to go back to take a look at this, because I'm thinking, I haven't made the final exam yet, but I'm thinking some of the final exam may come from here. The reason I think this is important is when you write an NIH grant, okay, you always need to do the justification, how many resources do I need, and how many things do I, and most of those needs to be based on the preliminary data. So based on the preliminary data, you can already generate this type of saturation point, right? And then this will help you to further propose additional experiment. How many rays do I really need based on the data we already have? So think through it. I know you haven't got it, and it took me a while to get it, but think through it. You will get it, I'm sure. And additional notes about this information is a particular MSRER value does not imply all the true binding sites with this enrichment will be identified. It doesn't guarantee that. It only tells you further increasing the sequencing depth won't help. All right? So just to make sure this is the concept, don't get confused. All right? And next couple of slides, I'm going to just flash through several standard comparison of different peak binder algorithms. This is the paper that Hai found out, so that I just give you a few, just a very brief introduction of their results. So comparison of different algorithms, regardless at what content, it's pretty difficult. And because so far there's more than a dozen of algorithms has been published, and independent evaluation is desired. We cannot say we publish this algorithm and see we are doing so much better than X1 and Z. Those are really sometimes not really independent evaluation. They are not really fair. And so this independent evaluation is desired. And sometimes it's very hard to get benchmark data sets. So meaning that we don't really know what is the positive or what is the negative, and how can we evaluate which method is doing better or worse. And this is the paper published in PLOS ONE the last year, and to compare different peak binders. And they did design several criteria. The first one is they choose the data sets on protein with well-defined binding motifs, like NRSF or GABP and FOXA1. So we already, before people do the experiment, we already know their sequence binding sites very well. And this will help us to give us at least some idea whether we can target the right region or not. And they also trying to evaluate the sensitivity, which is the ability to find experimentally validated sites. The good thing of all these several factors are there's hundreds, sometimes dozens of validated regions already been, people use PCR type of study and already find out hundreds of binding sites. And those are really experimentally validated. So they want to see how sensitive different methods are, meaning that what's the percentage of those binding sites I already find out. And they also want to look for the specificity, which is very, very difficult, and which minimizing the false positives. This is difficult because there's a lack of true negative sites. And we really don't know where this protein doesn't like. And it's hard to find. And the information they use is the percentage of the fragments they identified containing the binding motif. So the hope is if it's identified and it has the binding motif, that is more likely to be true. That's why that they focus on several of the proteins the motif has already been well defined. The first result here is the quantity of peak reporters. Okay, these are the algorithms they compared. Okay, and the last one is not, but all the others are the algorithms they compared. So for different algorithms, you can see that for different bars, those are different proteins. And for different algorithms, the programs were run with default or recommended settings, and default strengthening levels are set differently. So you can see that some of the algorithms identified much more than some other algorithms. That's really annoy people, right? How do we know that which one is correct, which is not? But they did do the core peaks. Core peaks meaning those regions that has been identified by all the 11 algorithms they evaluated. And this represents about 75 to 80% of the smallest peak list. And they also did a pairwise comparison of any two different algorithms. They found that 92% of peaks in the smaller list were also in big list. And about 45 to 50% of peaks in larger lists were also in smaller lists. I don't know how to make the use of this data, but the point here is just to be precaution, just like how just one or two algorithm tells you this is a peak or not, doesn't really give you a lot of the information. And these are, so at this stage, it's hard to evaluate their functionality. And sensitivity is a good one. So they use the qPCR verified true positives and high confidence motifs, and they want to see how many, what's the percentage of true positives, the positives they can recover. Okay, so you can see that most of these algorithms for this NRSF qPCR results, they are pretty similar, right? So after the peak ranking about 1,000 ones, they recovered almost 90% of the experimentally validated peaks. That's really, really good. And this one is start to separate a little bit. So you can see this is this is 11 different algorithms. So this is the GABP. So some of the algorithms, once you go to the let me do this. 5,000, you got about 80% of the reads experimentally validated ones that identified. But sometimes at the same number of reads, you got only 55% of identified. So this really gives you the sensitivity of different approaches. Let me give you the answer who is the best one. And the best one here is the peak-seq. I think it's by the Mark Gersten's group at Yale. Okay. And these two are based on PCR-based assays. This is the confidence on the motifs, whether this has the motif in it or not. But you can see these different trends. And another thing they want to look into is the peak accuracy, which is the percentage of peak with this motif within 500 base pair of identified region. So you can see this with the number of ranked peaks, and of course the smaller the number is, the higher the ranking is. And you can see that the higher the ranking ones, the percentage that have the motif is much higher. Once your ranking become lower and lower, and this percentage that have that motif become lower and lower as well. Okay, but nevertheless, this will be one paper you want to look into a little bit at the time that you choose different algorithms. Okay. All right. Now let's go to the next topic, which is the motif finding algorithm and the related theories. Let me get it started about 10 minutes, and then we can have another break. Okay. The reason we are doing this is once you do the ChIP-seq experiment, you identified so many genomic regions. You know this protein is binding in there. And then what's the next step? Right? Next step, natural step is you want to look into the sequencing features of these rays and to see the regions you identify to see whether there's a true binding sites or not, and how do you want to design further experiment on that. And we'll give you an example and how do we use this information. But before we go into that, I want to introduce a couple of theories related to the motif finding. This is not sequencing specific, but this should be very useful. So a practical biological question here is, if we know the DNA binding sites of a transcription factor, can we predict its binding sites in a given string of a sequence? Let's say this is the gene, the promoter region, this is the promoter region, and we know this transcription factors likes to bind on CCACGT, all right? And we know the promoter sequences, and can we know that whether this protein binds in this promoter or not? Okay? It's pretty simple. You just look for whether there are CCACGT in the promoter region, and then you should be able to get the answer. Okay? If there is one, it has the opportunity to bind there. But sometimes you look for this one. This is ACGTCCGG, all right? But the reverse complementary one of this, it will be CCACGT. And there's an option that this protein will bind there as well. And so the methods to do this will be paste and copy the promoter sequence into the word, and just search for CCACGT and the reverse complementary one, and then you will find the potential binding sites. All right, that is bioinformatics as well, right? And this is truly based on the sequence feature of this protein. But things are never that straightforward. We know transcription factor binding sites, they are kind of degenerate. What that means is we are thinking that this position 1 is A and so on and so forth. But sometimes, if this location is C, so this protein likes to AGAACA. But if this is a C or this is a G, this protein can also bind there. Okay? Not as well, but still can bind there if there's one nucleotide differences. And for this, most people describing this will use another terminology. If you read the literature, so it's called consensus, which is just to use the largest number of occurrence of that nucleotide there. And so this is still AGAACA. But sometimes you got this tied situation, which you may get into this. If two letter ties and you got A, so I'm sure that you read some papers and on the binding sites you will see A, and this is either CLG and AA, and this is either CLG and this is either ALT. So then, and this consensus sequence, this is how the people present the binding sites in old days, right? And but the problem of this is the consensus sequences are not suitable for very complicated patterns. For the reasonably complicated patterns, it's okay. Once we got to the real deal, and the data will look like this. Yeah. So if we see the same question, this is position 1 to 6, and this is the ACGT and all of the numbers. The consensus is AGAACA, but another presentation of this factor will be called the position weight matrix. Okay? So the number here is very simple. It's just a percentage of this the real binding sites that people experimentally determined that contain this particular letter. So you see 100% in the first position is A, you'll put 1 here, right? And the 14% is C, which is 1 out of 7 is C. You put 14% here, and the rest is G. So you just put the percentage there. And this is called a position weight matrix. And this is a better representation of a sequence binding size. Okay? And most people are using this, actually. And now we have to put that into a more probability framework. So the biological question here is, now I know this position weight matrix of this transcription factor. How do we assign a score to any particular sequence that describe their level of similarity? So meaning that this is my binding size of position weight matrix. By the way, this is not difficult to get. All right. So there's a database that has thousands of transcription factors, and there's position weight matrix are all in there. So it's not difficult to get. So the question here is, if I have a sequence which is ACAACA, and how do I know whether this sequence will be, how similar to that position weight matrix is? So the way you do that is quite simple. So you got ACAACA, you just ACAACA, point out those numbers out, okay? And then you calculate the probability that if this is the binding sites, and what's the probability I'm going to see this particular sequence? The way you do that is just to time everything together. And that is the probability. If this is the binding sites, I'm going to have 10% of the probability to observe this particular sequence. Okay? And another thing that I want to calculate, what is the probability that this is not a binding site? What's the probability this is not a binding site? If this is not a binding site, it's a random sequence, then I have 25% of possibility to observe ACGT in every single nucleotide. And then your probability time together will be this tiny, tiny number. Okay? And then you want to calculate the odds ratio of this is a binding site and not a binding site, and you want to divide these two, and you found this is 400 times more possible to be a binding site than not to be a binding site. And then what people do is assign a score, which is log 2 of this odds ratio, which is 8.72. Okay? Does that make sense? All right. Now, people think it's too simple, and let's make it more complicated. And there's another definition. It's called a position-specific scoring matrix. Okay? So what happens is, let's go through this again. And these are the numbers of, based on the existing experiment, and the numbers of different locations. And so for this one, you can see this is a P. It's very specific in this location, but it can be something else as well. And so this is the position, the counts. And then once you get to the percentage of position weight matrix, and then this part is 80%, 4%, 9%, and so on and so forth. Okay? So this is the position weight matrix. And if we do the calculation, we can use the previous slide method to do that. But people will convert this position weight matrix into another thing called a position-specific scoring matrix. So what that is you use this number divided by QA, which is a random, like if this is not a binding site, what's the probability you'll see an A? Usually everything is background is 25%. So you use 0.04 divided by 0.25, 25%. And then you do the log 2, you put the number here. So you can see that everything negative here, that suggests it's a lower chance comparing even to the random. But every single location, you will see a positive number there, and that suggests that this is represent the specificity of this particular binding sites. Does that make sense? But actually, I was talking about 25%, and actually that's not what people are using. People are using A and T, they use 30%, C and G, they use 20%. Because in the human genome, we have the AT accounts for about 60%, and CG accounts for 40%. But anyway, this is the percentage of this latter to be occurred in by random. So you can see this is matrix is called position weight scoring matrix, position-specific scoring matrix. But the question is, why we need this? Why we want to make it so complicated? Well, actually, once you derive this, the calculation becomes really, really simple. Okay, for this particular one, let's say calculate the matching score for TAT, AAT. So what you get here is you just circle out these numbers, add them up together, and that will be your score. Okay, so you just basically you transfer those previous odds ratio and the probability ratio back into here, and just make it just very simple calculation. And that's how you do this. And of course, you can calculate for any other binding sites, you can calculate this score as well. That makes sense? All right. So when you read papers, and you got another type of representation, which is the sequencing logo. So we started from this one, which is the position weight matrix, either the counts or percentage, and position weight matrix, a specific scoring matrix, the log ratios. This is for the ease of use. And when you read the papers, when you see, you have an idea what the binding site should look like. And when you present in this way, that's really annoying. But this is a much better way. So it tells you for this location, position, and it's better to be a G. And for this position, it can be a G or A or U, right? So this is called a sequencing logo. So basically, if you put your sequence into the software, they will generate this for you. It's not very difficult to do. And how to interpret this logo is there's two factors I want to consider. The first one is the total height for each location. And the total height represents the information content at a specific location. So meaning that for this binding size, this particular loci is very, very important. Okay? Don't mess up with that location. If this is a G, make it a G. If it's anything else, and probably the protein is not going to bind there, because this particular letter is so important. So for each individual total height, that suggests the information content of that particular location. And the height of individual letter is the contribution of that particular letter. Okay, so you can see the total height of this is this much, but total height of this is this much. That means for this particular location, you can mess up with it. If it's a G, you put an A, that's okay. You put a U, that's okay. Just don't put C there, right? So that will kill your specificity. But anyways, so this is how you read this sequencing logo. Now comes to this. How are we going to use the ChIP-seq data? How can we use this in the ChIP-seq data to study the binding sites? All right, let's take a 5 minutes break, and we'll come back. I see people are start to get sleepy.